These microscope images are of a healthy person's lungs. Note all of the air sacs or alveoli. The alveolus, the air sac, is the functional unit of the lung. Each lung contains about 150 million of them. And the alveoli are lined by simple squamous epithelial cells, very thin cells. And because they are so thin, and because the cells which line a capillary, the endothelial cells, are also simple squamous epithelial cells, very thin, then this respiratory membrane, this wall which separates the air in the air sac from the blood in the capillary, is so very thin that diffusion can occur. Oxygen can go from the air sac into the capillary, carbon dioxide can diffuse from the capillary into the air sac. If you compare those images of normal lungs to these images of someone who had viral pneumonia, you can see the problem. The alveoli are not clear so that they could have gases which could diffuse back and forth between the capillary. Instead, they are full of debris from dying cells and by all of the immune cells which have been attracted to the site of the infection. Immune cells, in addition to damaged cells and connective tissue cells such as mast cells, send out signals which attract immune cells to leave the blood and to enter the air sacs. But clearly this then would interfere with gas exchange. Part of the immune response is inflammation, and thus fluid will collect in these air sacs. Once again, this will then increase the distance between the blood in the capillary and the air in the air sac, and then limit the oxygen and carbon dioxide which can diffuse back and forth. And so by adding more cells and cellular debris and fluid and mucus to the air sac, someone with viral pneumonia would now have trouble performing the gas exchange in their lungs, which would be needed to support their body's tissues.